Hello, my name is Allison McGrath. The date is 11/17/2021, and today I am going to be discussing the regulation of sperm maturation in germinal epithelium. So, what is sperm? A lot of people know this, but it is the male gamete. So it fuses with the female gamete, which is the egg, and it produces a zygote once that formation occurs. So, sperm is here, the egg is here. So, sperm maturation is a in-depth process. At birth, the testes have immature sperm, and the sperm don't begin maturing until puberty stimulates this. Puberty specifically stimulates mitosis in the germinal cells, and these germinal cells are otherwise known as spermatogonia. The spermatogonia can take two paths in their maturation. They can either continue to undergo mitosis throughout a male's reproductive years to produce more germinal cells, or they'll transition into meiosis, in which they will first produce a primary spermatocyte. So the spermatogonia that transition into meiosis are what are responsible for ultimately producing sperm. So from the primary spermatocyte, another round of meiosis occurs, and then the secondary spermatocytes are yielded, and then ultimately following a pretty similar process, the secondary spermatocytes will yield spermatids, which will finally mature into sperm. So as you can see from the diagram in this image, one spermatocyte can yield 4 sperm cells. So puberty is directed by various control pathways. So in both males and females, the roles of gonadotropin-releasing hormone play a very similar role. So gonadotropin-releasing hormone is released from the hypothalamus, which in turn results in the release of follicle-stimulating hormone, which is FSH, and luteinizing hormone, which is LH, as well as activin being released from the anterior pituitary. So more specifically, FSH initiates and maintains gametogenesis by increasing ABP, which is androgen binding protein. And then LH acts on endocrine cells to stimulate production of testosterone in Leydig cells, which I'll get to what those are more specifically in the following slide. And then activin, you can think of it as activating spermatogenesis or promoting spermatogenesis. So in males, the feedback pathway is negative. In females, it's a little bit more in depth. Sometimes it's negative, sometimes it's positive, but we're just focusing on males right now. So this schematic right here is outlines the process pretty clearly. So you can see that the hypothalamus will release gonadotropin-releasing hormone, and as a result, it will act on the anterior pituitary to release FSH, LH, and then also activin, which isn't in this diagram. These hormones can, in turn, in a female, they'll have an effect on the ovaries, but we're focusing on males right now, and they will have an effect on the testes. Ultimately, as a result, the testes will produce testosterone primarily from those Leydig cells, and this androgen testosterone, as well as another molecule known as inhibin, will have a negative feedback role by inactivating the anterior pituitary and also inactivating the hypothalamus. So inhibin is it's kind of like the antagonist of activin, and it's released by what's called Sertoli cells, which I'll also get into on the next slide. So inhibin, along with testosterone, will allow for that negative feedback loop by inhibiting the hypothalamus and in turn the anterior pituitary. All right, so this slide, I'm just going to move myself over here out of the way. So this slide is kind of the meat of this topic. So the regulation of sperm maturation. So within the testes, there are tightly coiled seminiferous tubules, and between these coils are the Leydig cells that we were talking about earlier. They're positioned between these coils. Within the seminiferous tubules are what we mentioned earlier as the Sertoli cells, as well as spermatogonia. Now, the Sertoli cells have a basal membrane and an apical membrane. The basolateral membrane is where these immature spermatogonia initially reside, and then the apical membrane faces the lumen of the seminiferous tubules. And this is also known as the blood-testes barrier, similar to the blood-brain barrier. So as mentioned earlier, the Sertoli cells release inhibin used in the negative feedback, and they also release something called ABP, which was also mentioned earlier, which is androgen binding protein. And this is basically used to increase testosterone retention in the testes by making testosterone less lipophilic. So when stimulated by follicle-stimulating hormone and luteinizing hormone, which remember are initially stimulated by gonadotropin-releasing hormone, some of the spermatogonia are going to take the path of undergoing more mitosis to produce more germ cells, but some of them are going to take the path of undergoing meiosis in order to ultimately form mature sperm. So the spermatogonia become spermatocytes after one round of meiosis, and each round of meiosis is going the spermatogonia they're going to mature and move closer and closer and closer to the apical membrane, so closer to the tubule lumen. And then by the time they reach the luminal ends of the Sertoli cells, they will have undergone 2 rounds of meiosis and are now what are considered spermatotids. However, when androgen levels become too high, so this includes testosterone and also, as discussed, inhibin, the negative feedback loop is initiated in order to reduce this process of sperm maturation by ultimately reducing gonadotropin-releasing hormone release and then subsequently FSH and LH. So this is just a better schematic of what I was discussing earlier. It just makes it a little bit clearer when discussing the basal lamina and then the apical membrane. So let's see. So we have the spermatogonium here, and then surrounding it is Sertoli cells. So you can see the basal lamina labeled on the outside in this blue color, and then the lumen in the center, and you can see the more mature sperm in the lumen. Sperm aren't fully mature until they are immersed in a couple of secretions produced by some glands in the male reproductive system, but they're pretty close to becoming mature at this point. So you can see that the spermatogonium, they're going to undergo multiple divisions, and once they reach that lumen, they're going to be, one spermatogonium will become 4 spermatotids. And then you can also see these Leydig cells surrounding the Sertoli cells, and as mentioned earlier, these Leydig cells are going to be producing testosterone mainly. So that is all I have for this presentation discussing the regulation of sperm maturation. I will also be uploading a study guide, which is going to go through this processes more similar to the image on this slide, labeling the different phases of sperm maturation. All right, that is all I have. Thank you.